Today we are going to interview Professor Briana Burton. Her lab studies DNA transformation. Bacteria share many things in common with you at the molecular level. However, how they get their DNA can be very different. The DNA holds the protein recipes that created and maintain you and are inherited from your parents. We call this mode of DNA acquisition vertical inheritance. Bacteria can get their DNA from their parent cell in a similar process, or they can get it by other means. That's right, they can grab DNA from other places and modify their own chromosomes. This process is called horizontal gene transfer. One area that Professor Burton's lab works on is understanding the mechanism of transformation in a bacterium called Bacillus subtilis. Transformation is one of three modes of horizontal gene transfer in bacteria. So let's talk a little bit more about how it works. In transformation in B. subtilis, DNA is captured in the environment by a pilus. A pilus is a proteinaceous fishing rod the cell throws out into the environment. Once captured, the pilus retracts and brings the DNA to a transport complex that pulls the DNA across the membrane, eventually incorporating it into the chromosome. Scientists have worked out the mechanism of transformation in detail, but surprisingly, the pilus in action has never been visualized in Bacillus subtilis. Detecting it is no easy problem, and Professor Burton's graduate student, Jason Zucchi, used a clever method to label the pilus so that you could see it in a microscope. So what was that clever method? A pilus is made up of protein units expressed by a gene called COMGC. These COMGC proteins then assemble into a long rod that makes up the majority of the pilus. You can see the COMGC protein as its structure listed on the slide. The trick was to engineer the COMGC gene and insert extra cysteine residues. And a cysteine residue is an amino acid that has a sulfur element on it. By having these extra sulfurs in the protein, they could label it with a dye that sticks strongly to sulfur. You can think of it as them sticking extra hooks on the protein and then hanging reflectors on those hooks. When they shined a UV light on the proteins in the microscope, the pilus lit up and the scientists could see it wherever it was in the cell. They were able to follow the pilus in action, seeing it capture DNA and bring it into the cell. You will see this movie play during the interview. So why should you care? This work increases our understanding of how DNA transformation works in bacteria. Understanding transformation helps us to learn more about how bacteria evolve. They evolve rapidly, and because of this, become resistant to antibiotic treatment at an alarming rate. Transformation has the potential to spread these antibiotic resistance genes around, including to bacterial pathogens. The more we can learn about how transformation works, the better equipped we will be to counter the spread of these resistance genes and keep antibiotics working for longer periods. Hi, we are here with Briana Burton to learn about her work and why she's a scientist. And in fact, that is the first question. Why did you become a scientist?
I wish I had some really profound sounding answer for that, but I don't. I think it really comes down to the courses that I had both in middle school, high school, and in college that made me able to explore questions that I had and be curious and find ways to go answer those things. Sort of the creativity that's involved was something that was really great. But I could have gone into music or literature. I think it was just a sort of, in some ways, luck of having great teachers along the way.
So the teachers inspired you and got you excited about microbiology?
Definitely.
All right, yeah, cool. What is the broad area of inquiry that you work in?
I think in the field, most people would refer to the work that I've done and that my lab does as sort of structure function work, so broadly falling into this class of basic science. where we think about how biological molecules, especially macromolecules like proteins and nucleic acids, interact to carry out their functions in a cell. So we focus more on that how question of how these things are working together, as opposed to thinking about clinical applications, for example.
Mm-hmm. Okay. And then, so like, from what I've seen on your website, the information that, the papers that you published, you're really interested in how DNA gets across membranes, it seems. Is that fair?
That is one of the major areas we work on, yes.
Yeah. So what are the other major areas?
So we have the two systems that are focused on DNA transport across membranes. That's how bacterial cells segregate their chromosomes during cell division, and often that happens at a cell division site where they would otherwise guillotine the chromosome, and clearly that's a bad thing, to chop up your DNA. The other DNA transport system we study is in peer-to-peer exchange within the bacterial communities, so how bacteria horizontally transfer DNA. In our case, we're specifically interested in natural transformation, which is where bacteria go and try to grab onto DNA that's outside of the cell and bring it in.
Okay, so wait a minute. So bacteria actually share DNA between themselves and will just take DNA out of the environment?
There are many ways that they share DNA. Some of them are active, where there's a donor that will actively donate DNA to a recipient. The version that we're interested in is one where the cells are actually scavenging the DNA from their environment, from their dead neighbors, for example, who have just spilled out all of their genomic contents, and now it's available for anyone who wants it.
Interesting. Okay. Why do you think your research is worth doing?
I think a lot of basic science, we don't always know where it will lead, but learning new things about biology is always important. There's so much that we don't know, and we don't know what we don't know until we find out what it is. From our department, we have a great historical example of Professor Tom Brock, who went and discovered several organisms out in the national parks that eventually led to our ability to do PCR through his identification of these thermophiles. So there are always ways that our research could have impacts in the future that we don't know about yet. A great example actually that happened to me recently during this past year. For my PhD, I studied a virus that infects bacteria, so a bacteriophage. And one of the things that was really confusing at the time was that the speed at which the virus could replicate its genome and make copies of itself and move itself around didn't match with how fast it would have to make the proteins to do that. So it wasn't clear how it was actually carrying this out. And what I discovered during my PhD was that the bacterial virus was actually co-opting the bacterial protein garbage disposal and turning it into a recycling system. And that's how it was able to actually carry this out faster, because it could recycle things as opposed to destroying them. So fast forward 25 years later, this year, a group that's been studying a CRISPR-based gene therapy potential for human cells that had been shown in vitro and in tissue culture to work reasonably well, but was limited by its efficiency, went back and used our finding that you need this recycling system to improve efficiency of this particular class of proteins. They added the ability for their CRISPR system to use that recycling system, and now the efficiency is at the point where they may actually be able to use this as a real therapy.
That's a great example of using like basic research that seemed kind of esoteric at the time. It was important and interesting, but it had a huge useful property later on.
Right.
Excellent. All right, great. So discuss now a recent finding that you're really interested and excited about.
So going back to this idea that bacteria can share DNA amongst themselves with their peers, one of the things that we have recently been able to do in the lab is actually visualize the appendage or the pilus structure that the bacterial cell extends out to use to grab onto the DNA and to pull it in. And so the bacteria that were originally studied for this process in the structure function system have been studied since the 1960s, and we've known that in theory this structure exists and is probably doing this, but no one has been able to actually visualize it and learn about its dynamics until this year, when my graduate student Jason Zucki was able to get the imaging and experiments to work to be able to see this, and now we can learn about how it's actually grabbing the DNA outside of the cell and pulling it in.
So he has images showing...
We have movies showing these appendages coming out of the cell. We have double labeled images where you can see the appendage and then in a different color the DNA, and then you can watch it come back to the cell.
Wow, that's cool.
So it's really fun. Yeah.
All right. Next question. What class do you teach and what is the subject?
So I teach microbiology 612, which is prokaryotic molecular biology, and it fits very well with all of these types of things I've just mentioned, which is trying to understand how proteins and nucleic acids, in this case in bacterial cells in particular, are interacting to carry out all of their biological functions.
Okay, and then you teach that to all the undergraduate students.
Yep. So there's undergraduates and graduate students in the class. Yes.
Sure.
Mm-hmm.
So thank you. To end the interviews, I like to ask a fun question. What are your hobbies and what other interests do you have outside of work?
So a lot of my time is spent doing things that my child is interested in, so going to cross-country meets and things like that. But in my own time, I also enjoy running and hiking when I can. Probably the two most unusual hobbies that I have. I weave, so I make rugs and placemats and all kinds of things on a large floor loom.
Okay, so you have a large floor loom at home.
I do, yes.
Wow, okay.
And I've been doing that for a few decades, making things. And then I play a string instrument called the kantele, which is a Finnish instrument that most people have never heard of.
So is that a large instrument or a small instrument?
This is the one that I play is a 15-string instrument that can sit in your lap.
Okay.
So,
all right. Okay, fun. Thank you, Briana, for doing this.
Mm-hmm.